Plant growth and development. So what is plant growth and development? We know the first stage of plant is seed. This seed first germinate. After germinating, this seed produces small plant. After small plant, this small plant converted into an adult plant. This systematic development of and growth of plant is known as plant growth and development. For this plant growth and development, different process or functions are involved. One is called photosynthesis, another is called respiration, and transpiration. These three major factors are involved in plant growth and development, with other factors that also involve in plant growth and development. So what exact other factor that involved in plant growth and development I am discussing now? Growth is a irreversible permanent increase in size of a cell, tissue or organ. This is the cell irreversible growth of cell, tissue or organ. This growth is most fundamental and conspicuous character of a living organism. This is a most fundamental and conspicuous character. Growth is due to the expense of energy. One is known as anabolic and catabolic process. Due to this process or expense of energy, growth will seen. So this is about growth. This growth in plants. So what is growth in plants? This is the most growth is seen in a plant is unlimited growth. In whole life cycle of plant, the growth is unlimited because of meristematic cell. What is meristematic cell? Meristematic cell is the cell that helps in dividing continuously, because these cells are helps in division and that do not contain hard cell wall or do not contain cell wall and contain protoplasm. This protoplasm continuously this protoplasm in cell continuously divide and grows. So here new cells are added to the plant body. If new cells are added, then plant also grows. If plant grows, then plant have known as growth and development. So this growth in plant is known as open form of growth. Important. This is open form of growth. And the growth of plant is two types. One is known as primary growth and another is known as secondary growth. So what is primary growth? Primary growth means the in primary growth elongation of plant along their axis. So what is elongation of plant along their axis? Please concentrate here. This is a plant. If the growth is vertical, means at axis level, called vertical growth. This vertical growth is example root apical meristem and shoot apical meristem. This is the known as vertical growth. And next is secondary growth. Secondary growth is the lateral growth. This is the lateral growth. This growth we see at vascular cambium and cork cambium. And also in secondary growth, increase in girth of organ. This is the example of dicotyledonous plant and gymnosperm. This shows secondary growth. Growth at cellular level. So what is growth at cellular level? Cell contain protoplasm. So what is protoplasm? Protoplasm means that contain cytoplasm and nucleus. This composition of cytoplasm and nucleus is known as protoplasm. If the increase of protoplasm that indicate the growth of cell, indirectly the growth of cell that indicate or directly that indicate the growth of plant. So next is how to measure the growth of plant. We know the plant are a growth, but how? What exact parameter that through we measure the plant growth? One is known as increase in weight, fresh weight, dry weight. So first is increase in fresh weight. Increase in fresh weight means this is the plant. Let's consider this is small plant. This small plant grows after some time. When we measure, we found the weight is, let first is 10 gram, after some time it have 100 gram. So we can, through this conclusion, we can identify the weightage increases. And next is dry weight. Dry weight, one plant, one specific plant of the plant, we can measure through comparison at exactly this plant. Next is length, area, volume and cell number. If this is a plant root, this plant root is lengthwise, that lengthwise increases, area wise increases, and the volume of the root that also increases. Also the volume increases due to the cell number. If cells are divides, if these cells are divides, then the plant also grows. So this is the all about discussion of how at cellular level and measure the growth of plant. Next is phase of growth. The phase of growth, that is meristem, elongation and maturation. So meristem means in a normal term the activating cell or active cell. If any active cell that found that mostly divides. And elongation means that enlargement. Maturation means that get a maximum size. After maturation that cannot grow. In maturation stage that gain a maximum size. So one by one I am discussing. First is most active cell. This is meristem are most active cells and dividing cells. Because of that contain active cells and protoplasm continuously divide. So it is dividing the cells rich in protoplasm, and it contain the nuclei of the cell contain larger nuclei. Cell wall is primary means starting of cell wall. In normal term cell wall is absent in normally, but here plasmodesmata abundant. Plasmodesmata main function transportation of water molecule. During we see in symplastic pathway the transportation of water molecule through plasmodesmata. Next is elongation. Elongation means enlargement of cell. This is the enlargement of cell, and it contains vacuolation. What is the function of vacuolation? It store more water and minerals. Next, new cell wall deposition. So in elongation phase we see this cell are composed of cell walls, new cell wall deposition. Next is maturation. Maturation means it gain, the cell gain maximum size. It gain maximum size. And finally the deposition of cell wall here is shows wall thickening. The wall of the cell that shows thickening. The protoplasmic modification. Protoplasm also modified in this state. So it is the last stage where no cell, the at stage the cell is not, the cell get maximum size. Next another concept, this is called plasticity. Plasticity is the, in here let's consider this is a plant. This plant, the plant follows different pathways, maybe that is environment or that is maybe environment or phase of life. Due to environment or phase of life that affect the structure of plant in a different way, this system or this process is known as plasticity. If the environment, one of the example buttercap plant, and phase of life that affect the plant structure known as cotton, coriander and larkspur. In this plant we see the phase of life that affect the structure of plant in different ways. Next is types of growth according to cell division. We can identify what exact the growth rate and how the cell divides. So these are two types. One is called arithmetic, another is known as geometric. Means here you see the cell, this is a cell, this cell divides. After division, one cell, here one cell and another cell, it form two daughter cell. In two daughter cell, one cell is constant and another cell is divisible. One cell constant, another cell divisible. The divisible cell further divide into two cell, and in second generation, here in this generation, one cell constant, another cell divisible. This is known as arithmetic cell division or growth rate. And here geometric growth. So what is geometric growth? Geometric growth rate is one cell that divides to another cell, that divide another two types of daughter cell, and two daughter cells are further divide, but in arithmetic we see one divisible, one constant, but in geometric that both are divisible. Arithmetic growth are due to mitotic cell division. It is also mitotic cell division. Arithmetic also mitotic cell division, geometric also mitotic cell division. And here we see only one daughter cell can continue to divide, but here two are both daughter cell divides. And next is other differentiate, and it shows mature. Differentiate means it is specified to a system. Or let's consider one of the in case of human, a stem cell. In stem cell that further combine divide and forming a skin cell or skin tissue. As like differentiate means it is specified to a specific system or organ or tissue. Next, in geometric we see initial growth is slow. First is initial growth, this is slow. After some time we see the rapid growth, this is the rapid division. This is the rapid division. Next, condition for growth. Different condition for growth. We know what condition that require for growth. Condition for growth require for plant development and growth. If low condition, if harsh condition, plant cannot grow properly. If the condition is more well and advanced, then plant grow, or according to optimal condition, plant grow properly. So what exact factor that affect the plant growth and development? First is water molecule. This water molecule that affect plant. So how this water molecule affect? Water provide turgidity. How turgidity? We know during a cell, let's consider this is a cell. If water enters to this cell, that provide turgidity. What is turgidity? Turgidity means that gives swelling of cell. And second is enzymatic action. In cell, different types of enzyme are present. This enzyme are activated by water also. Next, water that helps in osmotic balance, balance of plant. Next one, oxygen. This factor oxygen, that elimination of metabolic energy. We know oxygen involved in oxidizing food and different types of metabolic process. So it helps in elimination of metabolic energy. Another factor, this is called nutrient. This is the most important factor. This nutrient are two types. One is called micronutrient, another is known as a macronutrient. This micro and macro helps in increase in protoplasm, and that the main source of energy, so that plant can grow. Other factor like light, temperature, gravity that affect the condition for growth. So what exact how this light, temperature that affect on plant, I am discussing later. So first connect with me, understood the next topic. So another factor known as PGRs, plant growth regulators, that is natural or artificial. Natural and artificial both are help in the development and growth of plant. So this is the sequence of development. This is the sequence of development process of plant. The major sequence of the development process of plant is first is the here is the cell. Here this cell divides after division that elongate, known as elongation process. After elongation that differentiate. Let's consider this is the leaf stem cell. This leaf stem cell first divide, after division elongate, after elongate that differentiate into leaf, that differentiated into leaf, then after this leaf is mature end. So this is the process. Here concentrate everyone. This is the cell division. After cell division is due to meristemic cell. We know this meristemic cell or meristematic cell that helps in division mostly. Then this meristematic cell divisible, after divisible it differentiate into specific cell. Let's consider here I am writing leaf cell. Then after this cell elongate, after elongation that cell shows maturation. Maturation means it get a maximum size, means the cells of the leaf, let's consider this is leaf, the leaf cell is get a maximum size, means do not for maximum size. Next this is mature, this mature cell further enter into senescence. What is senescence? We know senescence is the nutrient remobilize of cell death part. Here is the cell, if the cell is dies that remove nutrient or that provide nutrient to the young cell so that this young cell can survive. Then after the senescent cell undergoes death. So this is the process of plant or the development process of plant, how from seed to that grows at death. This is the overall process of plant development. The plant growth regulators are also known as hormones. These are known as hormones. These hormones helps in different types of growth and development. So these are known as different hormones: auxin, gibberellin, cytokinin, abscisic acid, and ethylene. These major hormones that affect plant growth and development. In this NCERT or according to Neet, we study these hormones. So one is this hormones helps in growth promoting activity. The major hormone that helps in growth promoting activity: auxin, gibberellin, and cytokinin. What a growth promoting activity? The growth promoting activity that lead finally the growth and development. One is known as cell division. If cell are divided, plant also growth. If cell are enlargement, plant shows also growth. And pattern formation. Pattern formation means we see here in leaf different patterns are formed. In one of the example in leaf, this is known as pattern formation. In tropic growth, what is tropic growth? Tropic growth means we know in one of the case the plant towards light, known as phototropic. So tropic means towards any condition. Another is flowering and fruiting in a seed formation. Flowering, fruiting, seed formation also helps by these hormones. And other is wound and stress tolerant hormone. This is known as one of the stress tolerant hormone is called ABA. Growth inhibiting. One is called dormancy and abscission. What is dormancy? Dormancy means this dormancy condition seed are not germinate. Another is known as abscission. So what is abscission? The natural phenomenon at which the fruit leaves are separate from the plant. Fruit leaves, these are separated from the plant. This dormancy is done by one of the hormone known as ABA. So detail we are studying detail about these hormones. So one by one. So first is auxin. This auxin affect different types of physiological effect in plants. So before studying about physiological effect, first studying discovery. First, it is discovered in coleoptile of oat seed, and this is as like auxin, a active compound that found in human urine. In human urine, one of the active component, let active component like the structure is as like auxin. So it is also known as a one type of auxin or compound found in urine. Next is auxin. It's IA. This is the natural auxin, and the synthetic auxin is NAA and 2,4-D. So what is IAA? Indole 3 acetic acid. Indole 3 acetic acid is the natural auxin, while NAA 2,4-D. NAA naphthalic acetic acid. This is the synthetic auxin, while 2,4-D is known as, that is a herbicide. Most important, star mark this point. This 2,4-D is the herbicide. And next is the auxin are mostly found. We know auxin, but where the auxin are found? The auxin are mostly found in growing regions. These growing regions are stem and root aphyses. This is the stem and, let's consider this is the stem aphyses, and this is the root aphyses, where the auxin are mostly found. The physiological effect of auxin. First point is xylem differentiation. So what is xylem differentiation? Differentiation or differentiate meaning one cell that differentiate. In previously I already discussed that's a one cell that differentiate into leaf cell. As like this xylem cell differentiate into xylem. Next, this another point of parthenocarpy. So what is parthenocarpy? Parthenocarpy means the fertilization without the fruit development without fertilization. In normally, the sexual reproduction occurs so that plant can develop into fruits. This fruit develop into a new plant. But in parthenocarpy, the condition is no fertilization occur, but fruit are developed. One of the example, tomato. This another auxin helps in apical dominance. So what is apical dominance? Apical dominance means we know this is a plant. This is the apical part of the, let's consider this is shoot, and this shoot contain apical part. Apical means the peak part. This apical dominance means that dominant over other part. What this apical dominance, that dominant over the lateral growth, of this plant shoot. So apical dominance is the main occur by auxin. This auxin help in different types of cell division. It is also help in rooting and flowering. So this is the all about discussion of auxin and with its physiological effect. Next point, another important point in auxin, growing apical bud inhibit lateral growth. Already discussed these things. So it is called apical dominance. So what is apical dominance? That inhibit lateral growth. This is mostly seen in, star mark this point, tea plantation and haze making. In this we see this apical dominance. Another hormone known as gibberellin. This gibberellin is mostly found, the first gibberellin found in one of the fungi known as Gibberellia fujikori. This is the active component known as gibberellin. This Gibberellia fujikori create a disease in rice. This is known as Bakane disease or foolish seedling disease or Bakane disease, in which the fungus affect the rice, and the scientists discovered this gibberellin is known as E. corsoi in 1926. Gibberellin have different physiological effects. So what exactly these physiological effects? So I am writing in a mnemonic way, A, G, B, R. So what is A? In A we study apple. This apple, if you spray gibberellin, this is gibberellin. If you spray gibberellin, the apple will elongate. After that, the quality also improve. Improvement of shape and elongation of shape, also quality improve due to gibberellin. Next is G. In G we study ganna or sugar cane. If you spray gibberellin, the length of sugar cane increases. This is the sugar cane. After spraying gibberellin, the length increases. Grapes. This is most important. Previous year question asked. Grapes. Grapes is length. If we spray gibberellin, that length of stalk, we know this is grapes. If we spray gibberellin, the length increases so that grape get space. If space, they grow more. It is also found in cabbage. If you spray gibberellin in cabbage, the cabbage growth also increases. Next is bolting effect. Gibberellin shows bolting effect. So what is bolting effect? Bolting effect means this is node to node of a plant is known as internode. This internode elongation is due to gibberellin. If I am spraying in anything, that anything means one of the example cabbage and beet, that shows bolting effect. Another is brewing industry is known as melting or GA3. So what is brewing in beer industry? If let this is wheat, applying gibberellin GA3, then that convert into helps in beer formation in beer industry. This is the all about discussion of AGBR. Now R, rosette. So what is rosette? If I am spraying, if the spray of gibberellin, that create a leaf-like structure known as rosette due to gibberellin. A flower, a leaf that become like flower-like structure. This is called rosette. So this is the all about discussion of physiological effect of gibberellin. Next is this gibberellin in different types. One is called GA1, GA2, GA3. Among all gibberellin, most active gibberellin is GA3 and mostly studied or extensively studied this hormone. This gibberellin, GA3 is mostly or extensively studied. More than 100 gibberellins are found. More than 100 gibberellins. So next hormone is cytokinin. So what is the function of cytokinin? Cytokinin is mostly two types. You see here, one is called two types of cytokinin found, gytin and kinetin. One is this is discovered first. Kinetin is discovered by herring sperm of DNA. Herring sperm of DNA at autoclave, and this DNA is this kinetin is the modified form of adenine. Everyone know what is adenine? This is the structure of DNA, adenine. This adenine also known as a purine. So this is kinetin discovered herring sperm of DNA. And gytin, yes sir, naturally cytokinins. So the natural cytokinins are mostly that is synthesized at a growing region, means the cell at the particular region that have high cell division at that particular place. This cytokinin are mostly found in this particular region of cell division are mostly root apices, developing buds and young fruits. At this particular region, this naturally cytokinins are found. Function. So what is the physiological function of cytokinin? It helps in producing new leaves, and that helps in formation of chloroplast in leaves. This is the most important point, lateral shoot growth. Due to, we know, this is a plant, this is the apical, and this is the lateral part. If the cytokinin increases, the concentration of cytokinin increases, then apical dominance block, while the lateral growth that's shown. If the cytokinin increases, apical growth block, and in another case we see the lateral growth that is occur. Another condition that is called adventitious shoot formation. Adventitious shoot formation. So what is this thing? Adventitious shoot formation means adventitious shoot is a kind of shoot not formed in its main location at the end of stem or internode. It is from the end of stem and internode. For example, if you take in the internodal part of explant, like this is a explant, this have capability to grow into a new plant. At this point, if we add cytokinin, after adding this cytokinin helps to grow adventitious shoot in this explant, in this the explants. So this is the example of cytokinin in adventitious shoot formation. Next is overcome apical dominance. We know if we add auxin, that helps in apical dominance, but if we add cytokinin, apical dominance block, so it shows lateral growth. Another thing is delay of leaf senescence. So what is leaf senescence? The nutrients are remobilized from cell death part to the younger part. In this condition is delayed by leaf senescence cytokinin. So this is the all overall discussion of cytokinin. So ethylene is the gaseous hormone that is known as, in the all hormone, only hormone that present ethylene, only gaseous hormone, and its physiological function. Already discussed, it helps senescence. What is senescence? I already discussed this thing. So it helps in senescence. Next is abscission. So what is abscission? Abscission means the natural detachment of parts of a plant, typically dead leaves, and ripe fruits. These are naturally detachment of from this plant. This is known as abscission. And next is fruit ripening. So how ethylene helps in fruit ripening? When ethylene added or naturally synthesized at fruit ripening or fruits, that increases the respiration. Due to increase in respiration, that create a condition known as respiratory climatic. Increase in respiration of fruit is known as respiratory climatic. What exactly happen? The exact mechanism happen here. Energy involved and cell goes towards death. So this is known as fruit ripening. Next is breaks seed dormancy. So what is seed dormancy? Let's consider this is a seed. If this seed is not germinate, this condition is known as dormant. If this is break the dormant, it is known as germination. So if we add ethylene here at a particular concentration, then this ethylene helps in germination or breaks seed dormancy. Initiate germination. Already discussed. If we add ethylene, that initiate germination. In germination like peanut, sprouting, potato tubers, that helps germination. Peanut, sprouting, potato tubers. A rapid internode and petiole elongation. So if we add ethylene, that helps in internode elongation. Internode elongation. Internode elongation in deep water rice plant. One of the example written, deep water rice plant. This is known as another point, synchronizing fruit set in pineapple. So what is synchronizing fruit set? So what is this means? Synchronizing fruit set in pineapple means in pineapple flowering and fruiting occur at the same time. This is known as synchronizing fruit set in pineapple. This is happen or this is induces by one of the hormone. This is known as ethylene. Most important. It enhances the respiration rate during ripening of the fruits. Also discuss this point. So in ethylene, one of the major component known as ethophone, that is widely used. This ethophone helps in fruit ripening, abscission and flowering. Fruit ripening like tomato, apple and abscission. Already discussed what is abscission. Only I am discussing example of ethophone that helps in abscission. One is cotton, cherry, walnut. In this plant, ethophone helps in abscission, and this ethophone are the major component of a ethylene. Flowering, that helps in female flower in cucumber. So this is the discussion of all about ethylene. Next hormone, this is abscisic acid. So what is the function of abscisic acid and what exactly the abscisic acid? That helps in abscission, means detachment of fruit set or leaf from naturally from the plant. Dormancy, this is major important point in abscisic acid that create a dormancy. If we add abscisic acid or AVA, that create dormancy in the seed inhibitor. So this is the inhibitor of plant germination and plant metabolism. Already know what is plant metabolism. Different cellular processor held by this abscisic acid, and see if we add abscisic acid that block seed germination. Abscisic acid that close stomata. If we add abscisic acid, that helps in close the stomata and act as a stress tolerant hormone. So how it act? We know through the stomatal difference. Through the stomatal difference, this thing happen. Next, seed development and maturation and dormancy, that is done by abscisic acid. This point is most important according to Neet prospect of view. If we increase AVA, then gibberellic acid decreases the consent. Means this hormone is the antagonist. It is known as antagonist. So what is the meaning of antagonist? Means let's consider this is a seed. If we add ABA, that block the germination. In case of you add a dormant seed GA, that helps in germination. So It is act as a VA and GA both are act as a antagonist. New concept, this is photoperiodism. So what is photoperiodism? Photo means light and periodism means what is the exact time of exposure. We are discussing about plant. So if I let this is a plant, this plant, this is light. Let's consider this is exposure time is 10 hour and another plant here, this is the exposure time is 5 hour. So 10 hour and 5 hour, assume that this time of exposure of light that affect flowering, that affect the flowering in plant. So why that effect I will discuss, and why flowering is need for new generation. If flowering happen, new generation of plant that occur, so it is need most mandatory for new generation. So light period and a dark period. In photoperiodism, plant goes or that realize two period. One is light period and a dark period. For flowering, only light period is not sufficient. It need to light period with dark period. So it is known as relative duration of light exposure. It is totally combination known as relative duration of light exposure. The point is one concept, critical time. Critical time means some plant that need a perfect amount of light. Perfect amount of light. If the light increases this critical time, this plant is known as long day plant. If this light less than the critical time, this is known as short day plant. If both are equaling flowering at this point, this is known as day neutral plant. So this is the discussion of photoperiodism. Next is vernalization. So what is vernalization? We previously discussed how light effect in flowering. Oh, flowering why needed? Flowering needed for next generation. So as like vernalization means how the low temperature that affect flowering. This condition is known as vernalization. So low temperature that affect the quality and quantity of light, quality and quantity of flowering. So the one of the condition, winter plant like wheat, barley, rye, these are the most important examples. Winter plant like wheat, barley, rye that shows this condition, and binary plant like monocarpic, sugar beet, cabbage, carrot, this shows this condition. So one of the thing I miss, that is vernalization. In vernalization we can do through one of the hormone known as gibberellic acid. That gibberellic acid helps in flowering at low temperature. Remember this gibberellic acid Ga act as low temperature and flowering. Most important, this helps in flowering. Another concept, this is called seed dormancy. So what is seed dormancy? Already discussed. Dormancy means that seed have no germination, that lose the capacity germinate only at this condition. So seed fail to germinate is known as seed dormancy. And why this seed fail to germinate? Different factor that affect seed to block the germination. To germinate, this is blocked by different factors or different things. So what exactly thing that block this seed dormancy? One is known as presence of chemical inhibitor. Hard seed coat. If the seed coat is very hard, germination not properly or that become dormant. Next is abscisic acid. This is most important. Abscisic acid that helps in seed dormant, that block germination. Phenolic acid, para-ascorbic acid, immature embryo. So what is immature embryo? If the embryo not properly developed, let's consider for the development of embryo that require 10 months. If it is developed in 5 months, this is known as immature state. Immature state. So it not get proper condition for maturity or germination. So this immature embryo cannot germinate or that held the germination. It is two types. One is called natural and man-made. So how we avoid this dormancy and helps to germinate this seed, create germination on this seed? We can do through these two methods. One is natural and man-made. So what is the natural? In mechanically, this is artificially. Artificially or man-made, we can remove the seed coat through different knives or different things. Next, natural. In natural, we do through the microbes. We know that this is a culture plate. This contain bacteria. This bacteria secrete enzymes. This enzyme that degrade the seed coat so that germination can happen. So next is passes through the digestive tract of animal. One of the example, let's consider dodo. Some plant seed that helps in germinated by dodo through their digestive system, when passes through the digestive system, and then after through the digestive system we can get this seed get a favorable condition for germination. Next is inhibitory substance. Inhibitory substance removed by one of the chilling effect known as chilling effect. If we add gibberellic acid and nitrate, most important, this point is most important, remember, inhibitory substance removed by chilling effect. Which effect? Chilling effect. The chilling effect created by gibberellic acid and nitrate. Gibberellic acid that act in low temperature also in flowering, that is vernalization, also discussed. So this thing, chilling effect, this chilling effect is created by gibberellic acid and nitrate. Seed dormancy removed by light and temperature change also. This dormancy also removed by light and temperature changes. So what is totipotency? This is first understood, then you can understood the what is dedifferentiation, redifferentiation and differentiation. First, the ability of a single cell divide and produce all of the differentiated cell in an organism. So what is the thing? This cell able to produce many cell, or this cell able to produce, let's consider this is a cell, this cell produce skin cell, this cell produce lungs, this cell produce liver, as a normal example. So this the cell, the ability of a single cell divide and produce all of a differentiated cell in an organism. Mostly spores and zygotes are example of totipotent cell. In case of plant, we see meristem cell. These are the totipotent cells. So for differentiated cell. So what is differentiated cell? Differentiated cell means this is a cell of meristem cell. We know this meristem cell have one activity that continuously divide, and the division of this cell that is differentiated at a specific cell. This specific cell, let's consider one of the example, root hair cell. This is leaf cell of mesophyll cell and a sieve cell of a phloem. So what is the function that gain? Root hair cell that gain the function absorption of water, but different from this meristem. One cell that divide into different cell, but this different cell have specific function. This is root cell, this is mesophyll cell, and this is sieve cell. The function of sieve cell, transport of food, while the function of this cell, photosynthesis, because it contain photosynthetic pigment. So next is differentiation some points. Permanent change in size due to differentiation. The cell become gain permanent size. Biochemistry changes structure and function of cell, tissue or organ. That may be all thing changes. The cells derived from meristem differentiate and mature to a perform specific function. So what is it? It is perform specific function already discussed in meristem. This meristem gain a root hair cell, phloem cell and mesophyll cell. The structure and function are varies. So next is de-differentiation and re-differentiation concept. So here is the carrot. This carrot already differentiated into a carrot. But if we take this differentiated cell in a test tube, let's consider this a tissue culture technique. If we take this cell in a test tube, then after this test tube, from this test tube grown in nutrient media, after growing this on nutrient media, this is called callus. This callus further transport into hormone media. This hormone media with as favorable condition give to this callus so that can grow. This callus can grow into same cell. This is a new carrot. So here we see already discussed this carrot, new cell taken, after this cell convert into new carrot. That means carrot gives a carrot. So this process is known as redifferentiation. So we confused in this differentiation, de-differentiation and a redifferentiation. So this is the overall topic of plant growth and development.